Hello, IB Bio classes. Welcome to part 2 of 3.4 Inheritance. So, we did go over a lot of vocabulary in the last video. I would like you to review that on this image here. So, would you be able to pause your video and see if you could maybe copy this into your notebooks and then identify what each of these lines is pointing to? If you said all of these things, if you were able to annotate the image like this with the labels and the brief descriptions, then you were able to successfully complete that. I would suggest just repeating what I just asked you to do over again without looking at the definitions before you move on with this video. So what we have here is homozygous dominant genotype of the alleles right here at the top. This is a homologous pair of chromosomes, right? So you have one from the mother, one from the father, and you could imagine that this is inside of any one of your cells. So inside of one of your cells, you do have a homologous pair of chromosomes. One of the chromosomes is from your mother, one is from the father. Now, are these replicated chromosomes? No, they're actually not. You may think, oh, this looks like an X shape, though. You said that that means that it's replicated. It's not, though, because they're not connected right here at the centromere. So they are separate chromosomes, and they're separate unreplicated chromosomes. So on the mother's chromosome, at this locus right here, she has a dominant allele, and the father's homologous chromosome that was given to you also has a dominant allele. So your genotype would be homozygous dominant for that particular trait and for those alleles. Over here we have a homozygous recessive genotype for a different trait, and that's represented by two lowercase b's. Okay, as we move on, you can see the centromeres right there. Remember what genotype is. Genotype is the combination of two alleles. The phenotype is the expression of those alleles. So you would see an expression of what right here? You should see the dominant expression of the alleles here. Right here you would see the recessive expression of the alleles. If you had a heterozygous pair of alleles as shown right here, uppercase D, lowercase d, then you would be looking at a different phenotype. You would be looking at a dominant phenotype right here, and it would be the dominant phenotype as determined by this dominant uppercase D allele over here. Now, in the case of a recessive disease, for example, lactose intolerance, which we were just talking about in our other example, right? It is carried by the recessive allele, okay? This genotype here would be what? Heterozygous, which means there's an uppercase dominant allele and a lowercase recessive allele in the genotype. What would the phenotype be here? It would be expressed by this dominant allele because the dominant allele always covers up or masks the recessive allele in a heterozygous phenotype. If somebody does have a heterozygous phenotype for a disease, for example, if this was lactose intolerant, right, they would be considered a carrier, okay? And in fact, all of those offspring in that other example that I was showing before, if you rewind to the lactose intolerance example, the offspring and the children were all carriers because they were heterozygous for the recessive allele that determines lactose intolerance. That's the disease-causing allele. They're not actually expressing that allele, but they are carrying it. That's why they're called carriers. Their phenotype does not show that they have that disease, but in fact, they are carrying the disease. Now, if you look over here to the only thing we haven't talked about yet, you've got a codominant pair of alleles right here. And we're going to be talking about that later, so I want you to think about it later. Do notice the notation here. There's no uppercase or lowercase, there's just a letter with a superscript of another letter on each of these. And you can see they have different superscripts, codominant. This means that both of the alleles are actually expressed in codominant inheritance. This is a different form of inheritance. Remember, if we go back to the first slide, this essential idea here is the inheritance of genes follows patterns. So there are different patterns, and we'd only talked about classic Mendelian inheritance patterns so far. I'm going to do an example of classic Mendelian inheritance in a second, and then later we will talk about codominant inheritance patterns. So my example of classic Mendelian inheritance patterns is going to use pea plants. Why am I using pea plants? Well, because the founder of this inheritance pattern, Mendel, Gregor Mendel, actually discovered this inheritance pattern. So if we draw a Punnett square, and then we draw some alleles out here, I'm going to choose green and yellow for the color of the pea plants. So Gregor Mendel noticed that some of the pea plants end up with a dominant allele that codes for the color green. Others code for a recessive allele, that's a lowercase y, which would code for yellow. Okay, so let's say we have pea plants that are, the mother is, what kind of genotype would this be? How could we classify that genotype? If you were thinking heterozygous, you would be correct. This would be the father's genotype, lowercase y, or little y. That would be homozygous what? Recessive. Okay, now we're going to complete the Punnett square. By the way, notice that I'm always putting the uppercase letter first. So that is one convention that you do need to follow. If there's two lowercase, you just put the two lowercase. Okay? If there's an uppercase letter in the genotype, you do have to put that first. That's one of the conventions of Punnett squares and genotypes. Okay? So we can see that the ratio here, what is the phenotypic ratio here? Phenotypic ratio is going to be what here? Phenotypic ratio is actually a 1-to-1 ratio because we've got what? What's the phenotype of these heterozygous genotypes? We can see that that phenotype is actually green because they have that dominant allele, the uppercase Y, big Y. And there's two of those, right? And then we've got over here two lowercase genotypes that are going to code for yellow. Okay, yellow pea plants. And we can see there's two of those. So we're going to actually reduce that fraction, the 2-to-2, right? It starts off as 2-to-2, but we reduce it to 1-to-1. Just like in math class, when you want the lowest possible fraction, the smallest fraction possible, that's exactly what you're doing here as well. Genotypic ratio actually ends up being exactly the same in this example as the phenotypic ratio because we have 2-to-2 again with the genotypes. Now, let's say we did a different Punnett square. Let's say that we had two heterozygous parents. So we have uppercase, lowercase. We're going to do the Punnett square, and you would end up with something that looks like this. And so you can see that this ends up giving you a different phenotypic and genotypic ratio. Okay, so we can see that the phenotypic ratio is actually going to end up being what? It's going to end up being a 3-to-1 ratio because we've got right here a green, 25% green, 25% green over here, and 25% green over here. That's 3 to the 1 that is recessive yellow. Okay, over here we can do the genotypic ratio, which is going to be 1 to 2 to 1. And in fact, maybe you noticed from our previous example with lactose intolerance that I gave you, when you have two heterozygous parents, you always end up with a 1-to-1 ratio for the genotype. And for the phenotype, you will always end up with a 3-to-1 phenotypic ratio.